So I'm going to start. We have like approximately 2.5 hours. And my plan is to, so Chuck and Noam gave you some really nice introduction about chromatin and ChIP-Seq. I'll go very briefly on some scientific discoveries that are out there. We can revisit them later. And then most of the time, I want you all to go through the workshop that I've sent you and gave you some sheets for it. Thanks. And the good news is that the GenePattern seems to be working. I'll talk and we'll start working for the next hour or so, and then we'll take a break. And then during this break, I'll print out the GenePattern, which will be part 3 of what you're doing. And it will allow us to do some scatter plots and correlation matrices. So as Chuck described, one of the very interesting discoveries we had together with Oren and Noam in the past is regarding to chromatin regulators. And Chuck gave a really good introduction regarding what they are and what they do. And I want to kind of hand to you one of the very interesting things that we saw. So in general, if you consider chromatin regulators, they can be activating, for instance, adding an active mark, or repressing, removing an active mark, or the other way around, adding a repressive mark. One of the very interesting ideas that we've seen is sometimes on the same genomic loci, we see the activating chromatin regulator sitting together with the repressive one, as if you have the gas and the brakes on the same place, allowing perhaps some better fine-tuning. And we're following up on this and looking into understanding it better. And we also seen that it can be like the binding is combinatorial. And then like you have in different cell types and in different locations, different combinations of chromatin regulators that allow you to understand better, like perhaps allow the cell to better regulate expression and organization of genes. We also have a website for that. And another point, and Noam discussed it, that we just came from a really nice meeting from the ENCODE. This is from the publication of the ENCODE studies in half a year ago. And I encourage all of you to download. They have like a nice thing for iPad, or the entire website is for free. And one of the really nice things is that they try to connect the studies to within themselves. So you have like, okay, I want to look into to choose one, and then you can choose a paper, and you can choose, okay, which are the papers that are involved with this kind of identification of epigenomics, organization, and so on. And as Noam was talking a lot about enhancers, there were some really nice studies giving ideas and better resolution of what enhancers are and how are they organized. And one of the, like, and I know Noam has also discussed like the motifs, and this is using the DNA as one approach, which again we can revisit later. But from my perspective, one of the points that I love to mention on this is I did my grad studies in experimental biology, and when we talked about enhancers, I think there were known approximately 5 to 10 enhancers that most of them were kind of known, the people have discovered them because of mutations. So for instance, in the human beta globin, they've seen that when a chunk of the area upstream to the gene is deleted, even though the entire gene is intact, there's no proper expression of the globin gene. So up till this point where we can have some marks directing us to what enhancers are, and where they're located, and even to exactly what is the predicted motif for binding over there, it was really difficult to go over and kind of get a global kind of annotation of enhancers on the genome. So these are like 3 or 4 studies that I wanted you to go over. And now the way that I like to do it is I gave you the handout. I think that you can all just go and follow up on them. And if anyone has any questions or something is not clear, just raise your hand and I'll try solving it. The basic idea behind what I was trying to do here is to give you kind of a taste of the approaches that we use for analysis of chip sequencing data. And like I see some of you here are computational people, and so there are many tools for doing what is outlined here. And I would totally encourage everyone that has no computational experience to start kind of thinking of finding ways to get a better hold of Unix, because everything that is done in the work that is here is much more flexible and easy if you do it using Unix tools. The reason that I was doing it, that I'm doing it in the GUI, like the easy interface for usage, is that this way everybody can do it and can understand. And in general, one of the ideas that I had in mind in building it is that many of my biologist friends, the experimental biologists, when they do or when they want to look at on ChIP sequencing data, they kind of get stuck. It's like, okay, how do I analyze it? What do I do? And what I was trying to do here is to go from the point that, let's say, you get the reads from the machine to the point that you can visualize them and analyze them and so on and so forth. So please go and start going over the instructions. And again, I'm here, and just raise your hand if something is not clear in how to do it. But one of the things that is the trickiest for people that want to look at genomics, can you hear me? Is that, okay, you can look at them at ChIP sequencing data on IGV. And if you look here, this is approximately 90 kb. But the genome is much, much larger. And even when we look at it and we see peaks and we get ideas, sometimes we want to compare two data sets, or we want to compare more than two data sets. And one way to do that is to just take the genome, and for each of the data sets, what we can do is to, let's say that this is data set number 1, and this is data set number 2. We can take for each one of them all the reads coming from the ChIP sequencing. So as Noam and Chuck have described, the ChIP sequencing is kind of a fractionation way. We do the ChIP, we fractionate the genome to what is in association with the protein and what is not. And then we sequence the DNA pieces that were in association with the proteins. And what we get are reads falling on some areas of the genome. Now, when we want to compare between two data sets that have like this kind of genomic-wide distribution, what we can do is to break the genome into the genomic area into really like kind of small, what we call beans. So instead of looking at the entire genome at once, we look on each of the chromosomes. And within each chromosome, we take each one of them and take only, for instance, 1 kb or 5 kb beans or 10 kb, but much smaller than the entire chromosome, or of course much smaller than the entire genome. And what we have tried to do with GenePattern is to help you do that on a GUI, on an interface that you can use online. And the idea is that we can take data sets, and the scripts that are implemented into GenePattern will take each of the ChIP sequencing, which at this point will be BOM files that in the first part you should be able to generate it using Galaxy, or if you're more comfortable with the Unix environment, you can do it with Bowtie or BWA. And taking these BOM files, we are then going to generate what we call bin files, which are kind of vectors, where for the beginning, it's going to be for each 25 base pairs, we're going to have a score of how many reads are falling on these 25 base pairs. So we're going to have numbers. And then the next step will be to take these bin files and take a genomic area of interest. In the example that I've sent you, it were what we call promoters, which are the areas around the transcription start site. And we call it a bed file. And the bed is usually chromosome, like it's going to tell the script or the machine which chromosome you're interested in. So it's going to be chromosome, and then the start position and the end position. It can have more features, but these are the ones that we care about for now. And then using these two files, what we're going to get, together with a decision of what size of chromosome, what size of genomic areas we want, we can make, I mean, these can be, for instance, let's look on enhancers with like 20 kb around them. But we can look within these 20 kb on fragments of 1 kb or 300 base pairs or whatever we want, as long as we can even, this is like the highest resolution is 25 base pairs. And then taking these two parameters, we're going to get a file that will be per each region that we decided, which is decided here, and per the fragment size that we decide, which can be anywhere from 25 base pairs to whatever we want, let's say 1 kb, for instance. It's going to make kind of a matrix that will tell you, OK, on this chromosome location, you're going to have these areas, and they have the average or the max or the median score. Again, these depends on the parameters you choose in GenePattern. And we can make it for data set number 1 and for data set number 2. And once we have that, we can, for instance, do a scatter plot that will tell us, OK, are they similar on all these genomic locations, which each dot can be something like what we would view over here. But it's going to be all over the place. And we can also make a correlation matrix if we use more than 2 data sets. And just a second, again, what I've sent you, it should direct you to now a directory that already has the data. So it shouldn't take, there's no the problem of uploading, which usually is not a problem. And we'll try and figure it out. Yes, he was first. Yes.
And they're not pairs of sequences, they're just individual, like they're just a long list of individual sequences. Sorry, I just started up some mistake. Anyway, the point is, short sequences, you have a long list of them in each file. You have 2 files, and you want to compare them. My question is, how are you comparing them? And you drew a plot, like a scatter plot, and what were the horizontal and vertical axes?
OK. So thanks for the question. The scatter plot will be that for each, so this will be data set number 1. And this will be number 2. Sorry. And for each one of them, for each genomic location, we're going to ask how many reads, what's the average number of reads falling on this genomic location. So for instance, this will be the average number of reads. Let's say that when we take fragments of 5 kb, we're going to have fragments of 5 kb that has almost nothing. And then we're going to have fragments of 5 kb that has more in both of them. So this will be, let's say, I don't know, 10 reads per 5 kb, or average of 100 reads per 5 kb, or so on. We also do some normalization for 20 million reads in it, but I don't want to get it too complex. But the idea is that this will tell you, when I look at data set number 1 on a specific genomic location, and on data set number 2 on the exact same genomic location, did both of them got the similar number of reads, which tells me if they're comparable or they're distinct. But you do it for each genomic locations, and then you can really compare the pattern from the ChIP sequencing in data set number 1 versus the data set number 2. Yes?
Yeah, my question was about the normalization, and when you're doing the bed format, like the strand specificity is not important?
It is important, not for this case, but for instance, for the TSS table, the one that we use is when we took into account the strand specificity, and then we know if a gene going into this way or that way. But we're trying to simplify it to the point that it's easiest. But yeah, it's a good point. And just one word about normalization. The way that we're doing it now, since when you do chip sequencing, it's not necessarily that you're going to get the same number of reads in both instances. So you can do chip sequencing for sample number 1, and you're going to get 10 million reads, and for sample number 2, and you're going to get 30 million reads. In the GenePattern module that we've generated, we take each sample and we normalize it for what is the ratio for 20 million reads. So by the end of the day, it's going to be, if we had only 10 million reads, it's going to, we know that we have like half of the reads that we would expect. If we had 30 million reads, we're going to have 1.5 of the reads that we expect. But this way, we can compare the two samples and see, like, and we take into account the number of reads. And we take only, like, the number that we look into is the number of aligned reads. So if there is some noise in the system, it kind of helps us avoid it. Any other questions? OK, so I hope this one will be smoother than the other part. I'll also try to figure out what was going on there.
Yeah, I'm not seeing.
No, OK. Regarding the GenePattern, in what I've sent you, there is a link for the internal BroadE GenePattern. Click on this. It doesn't work?
No, it shows you.
And just before you leave, I want you to kind of give you a couple of, at least, the bird's eye view of what we were trying to do, regardless of the VPN and Galaxy issues. The idea was as follows. We took raw reads from a sequencer, or these raw reads were coming from, we downloaded them from the ENCODE consortium datasets. And then we used an aligner, which the aligner takes the reads and knows in a very quick way to find where they are located on the reference genome. So this creates a BOM file, which is just, it's called BOM for binary. And this just allows, binary makes it a bit smaller in terms of size that it is on the server. And the file, it tells you where on the genome each read was located, along with some other parameters such as the quality of the read and so on and so forth. Later on, we can create a SUM file, which is identical to the BOM file. It's just a text file. And using these, we can do some kind of modification on it, such as when you take the BOM file, you can make a visualization file out of it, which is TDF, or some of you are familiar with BigWig or WIG file. And these files can be then visualized using what we were trying to do on the workshop was IGV, but you can also use BigWig, for instance, to visualize on the UCSC browser if you like it better. And also, once you've got the BOM files to work, I hope that the module that we've been working with GenePattern to implement will be available not only for the internal, the VPN only approach, and then you can, for instance, normalize by wholesale extract and take into account how many reads were in each file, and create a matrix that compares several data sets and compares them throughout the entire genome. So I'm going to be here till 5, but I've just seen that people start getting too upset about all the VPN and other issues and Galaxy. Probably I should have notified them that 40 people will try to use the website. But again, if you have any questions and any issues that I can try to solve, I'll be happy to assist. And you can also try it later on. Perhaps when you do it at home, it will be less usage of the Galaxy. Yeah. I've sent you all a link for the GenePattern, and there should be another one that I hope is on the way. that will also, I uploaded it to my public directory, so it should be a link there. In the same directory that the data sets for BroadE are.
So this is only if you're having trouble accessing?
Yeah. Whoever had some VPN issues, all the people with partner login, now this one should work. And in case you can't see the internal directory where we put the stuff, I've sent you a link for the public website, a public website that has, yeah. But there's also, where is it? There should also be another one that that has some files that you can explore. It's just chromosome 19 for 2 biological replicates of, yeah, it's called, instead of data, it's called GenePattern. Just go up directory and you see their GenePattern subdirectory. I can come and show you, but it's also, you know what, I'll just show it on the, okay, so if you go to the link that I've sent you before, there should be an email, but sometimes these emails take a bit longer. I just made a directory, and I named it GenePattern, and in it these 2 biological replicates, the chromosome sizes, which in a second I can explain why you need it, and the bed file that is the transcription start site table. So this should all be in the public website in case you can't log into the internal one. In order to bypass the Galaxy issues, if you go to broadinstitute.org /A Goren BroadE IGV, I put here the BOM files with the index, which is required to run IGV, and you can download them to your machine and run IGV tools on it. And then with IGV tools, just follow to the point where it tells you how to run IGV tools and create the TDF, which is part 6.
So we're launching IGV from the space?
It does. It's not necessary. You can just go to IGV itself, but just use the software, which the software can be done like this.